Perceiving where light comes from is vital for plants, helping to maximize their sunlight intake required for efficient photosynthesis. But with no visual organ like humans, how do plants know where light comes from? A recent study published in the journal Science has uncovered a surprising feature within plant tissue: air-filled channels that help them detect the direction of light sources. During photosynthesis, plants convert energy from sunlight into chemical energy. This chemical energy is critical for life on Earth, and as primary producers, plants are responsible for the production of nearly all the food we eat. By sensing the direction of light, plants can optimize their light absorption through a phenomenon called phototropism. This involves redirecting the growth of their shoots towards a light source. Specialized photoreceptors, known as phototropins, have long been known to aid this process, with the gradient of phototropin activation determining the direction of growth. What this new study finds is that air-filled intercellular channels between plant cells can help enhance this gradient, improving a plant's ability to detect directional light cues. According to the lead researcher, Professor Christopher Fankhauser, it all started with the observation of a mutant of the model species Arabidopsis thaliana, the thale cress, whose stem was surprisingly transparent. When these mutants failed to respond to light correctly, researchers decided to investigate the differences in the optical properties of the mutant and wild-type species. We found that the natural milky appearance of the stems of young wild plants was in fact due to the presence of air in intercellular channels precisely located in various tissues. In the mutant specimens, the air is replaced by an aqueous liquid, giving them a translucent appearance, says Fankhauser. But what is the importance of these air-filled channels? Well, they allow for the formation of a light gradient so that the plant can locate the light source. This phenomenon is due to the differences in the refractive index of air and water, with the latter making up the majority of living tissue. The refractive index refers to the speed of light when it changes medium. This change in refractive index between the water-filled plant cells and the air-filled intercellular channels leads to light scattering as it passes through the seedling, producing a light gradient that can help determine where the source of light is coming from. These air-filled channels have also been found to play numerous other roles in plants. For example, they can help promote gas exchange and reduce the risk of hypoxia when there is a reduction in the quantity of oxygen during flooding. Further studies aim to investigate the formation and maintenance of these intriguing structures.